reductase then takes and reduces NADP plus to NADPH. So the electrons are moved to the electron carrier. Once photosystem 1 loses its electrons, it will then steal the electrons from plastocyanin, allowing this chain to keep moving. Now, we look at what happened over here with the electrons of chlorophyll A in photosystem 2, and these photosystems are capable of getting electrons from water. The electrons are then moved to water, and all of this takes place within the membrane, the thylakoid membrane. So, boost the electrons, pull them to plastoquinone, pull them to cytochrome B, pull them to plastocyanin. Light kicks the electrons of photosystem 1 up, and they get moved all the way over to NADP plus to make NADPH. And then the electrons from plastocyanin replace those lost by chlorophyll A in photosystem 1. And then photosystem 2 steals electrons from water to make oxygen and hydrogen ions. This oxygen is the oxygen that's given off as a waste product of photosynthesis. That's a waste product of photosynthesis. And this oxygen is the oxygen that we breathe. So when we talk about plants giving off oxygen during photosynthesis, that's the stuff we're talking about. Okay? All right. Now let's look in this process a little more detail here. So I have another version of this, right, from a different textbook. Okay? But it works the same way. Light, plastoquinone, plastocyanin, light over here to make NADPH, strip electrons off of water, and there you have it. Okay? Now, notice what's happening here at the cytochrome B complex. It's a little active transporter. Most active transporters you've talked about in MCB, these active transporters are using ATP energy to move something against its concentration gradient. In the case of electron transport chains, the energy that's being used is the kinetic energy of electrons. And that kinetic energy allows for the active transport of hydrogen ions. So this plastoquinone loses electrons to cytochrome B. As the electrons move through, we're pulling hydrogen ions into the lumen of the thylakoid and removing them from the stroma. Okay? So when I do MCB, I often talk about this, but energy transduction involves changing forms of energy. So if I change the light energy to kinetic energy of electrons, now I have potential energy of electrons, energy of position. Now I have kinetic energy of electrons. That kinetic energy of electrons is changed into kinetic energy of hydrogen ions. And now what I have here is a high concentration of hydrogen ions, which is a form of potential energy, energy of hydrogen ion position. The unequal position of the hydrogen ions is a form of potential energy. I've converted light into potential energy of hydrogen ions. Okay? Now, the next piece of this is a transport protein that I didn't draw in here yet, but is present in both of our images. In this image and in this image, there's a little ATP synthase that's present in the thylakoid membrane. Okay? This ATP synthase, let me just stick it over here, right? So let's put the reductase, NADP plus reductase, and then put the NADP plus here, NADPH, and then here, let's put ATP synthase. Okay? I have a high concentration of hydrogen ions in here relative to out here. So these hydrogen ions are going to move through my ATP synthase through this facilitated diffusion protein, ATP synthase. And as they do that, we will allow for the phosphorylation of ADP to make ATP. Okay? So I'm using the energy of the gradient to manufacture ATP. It's basically how the light reactions work. A couple of terms here. The first term, the splitting of water, is called photolysis, right? Using light energy to split, that's photolysis. And the way that this ATP is manufactured in the chloroplast is called photophosphorylation. You now know 3 ways to make ATP. Photophosphorylation is what's done during photosynthesis. In cellular respiration in the mitochondrion, there are 2 other ways to make ATP: substrate level phosphorylation and oxidative phosphorylation. So the 3 ways to make ATP: substrate level phosphorylation, oxidative phosphorylation, photophosphorylation. Okay? Notice that the ATP is actually produced in the stroma, and the NADPH is produced in the stroma as well. That will be handy because what we want to do is manufacture carbon molecules, triose, in the stroma. So we're producing the molecules that are used in the carbon fixation reactions. We're manufacturing those right in the place where they need to be, which is in the stroma. Okay? All right. I think I have one more chart in here of the light reactions, just different ways to represent the same thing. I'm not going to talk about this year, but let's go ahead and walk our way through the carbon fixation reactions. Okay? So I'm going to do the carbon fixation reactions as a linear pathway, a biochemical pathway. I'm not going to do it as a cycle yet. We'll do it as a cycle later. Let's just do it as a series of reactions first. So the first reaction here is we're going to start with a molecule called RUBP. The technical term for it is ribulose 1,5-bisphosphate, but the botanist didn't name it, so we abbreviate it. Right? 1,5 refers to the 2 carbons where you find the 2 phosphates. The ribulose means that it's a carbohydrate. OSE, O-S-E, means carbohydrate. So this RUBP is a 5-carbon sugar with 2 phosphate groups attached to it. RUBP. Okay? Now, RUBP can be converted into a 6-carbon molecule by adding a carbon from CO2. Okay? That 6-carbon molecule that's produced is 5 plus the 1 carbon found in CO2. And the enzyme that's capable of doing this is a molecule called Rubp Carboxylase /oxygenase. And again, we abbreviate this one as Rubisco. Rubisco is an interesting enzyme. It is the most common enzyme on the planet. It is found in every single photosynthetic organism on the planet, be it a bacteria, cyanobacteria, an alga like kelp, or an oak tree. I all use this same carbon fixation reaction. That is, this trait, we would say, is kind of fixed in organisms because that's the way you do it, right? You take a 5-carbon RUBP, add a carbon to it using Rubisco, and you manufacture a 6-carbon molecule. Okay? The 6-carbon molecule is extremely, extremely unstable. In fact, it doesn't last very long, and it's so short-lived that when Calvin first worked out these reactions, he didn't even notice the 6-carbon molecule. The 6-carbon molecule fractures immediately into 2 molecules of PGA, both of which have 3 carbons. When carbon fixation occurred, Calvin saw the 3-carbon molecule, so he refers to this type of photosynthesis, the most common type, as C3 photosynthesis, because the first detectable product of carbon fixation is a 3-carbon molecule. Okay? A 3-carbon molecule. All right, so far so good. RUBP plus a carbon gives me a 6-carbon that fractures into 2 3-carbon PGAs. All right, there's my PGA. They're just showing us one of these, but keep in mind 2 of these reactions are going on. Then what we're going to do is hit the PGA with a high-energy phosphate group. That high-energy phosphate group comes from ATP and generates what's called an unstable intermediate. Now they give this molecule a name. They call it 1,3-bisphosphoglycerate. I'm just going to call it an unstable intermediate. It doesn't last very long. Basically what we're doing is using the phosphate group to transfer energy to PGA, and as PGA gains that energy, it reorganizes into a higher energy state and then falls back down into a lower energy state. This lower energy state is a molecule called PGAL. And in order to assist with this, we need to take the electrons from NADPH to manufacture the PGAL. Okay? So I would do the same reaction down here. ATP gives me ADP, the unstable intermediate. NADPH, NADP plus, and I've got my PGAL. PGAL, like PGA, has 3 carbons. PGAL is known by another name. It's called glyceraldehyde 3-phosphate, and the animal people, the cellular respiration people, call PGAL G3P. So if you remember G3P from cellular respiration, PGAL is G3P. It just has 2 names. All right? Now, PGAL is triose. This is the molecule right here that the plant was trying to manufacture. Through this simple reaction, or sets of reactions, it manufactures the 3-carbon PGAL. This 3-carbon PGAL can then be converted into whatever you want. It can be manufactured into a glucose by taking 2 PGALs, or fructose or galactose by taking 2 of these. You can make a sucrose by taking 6 of them, or sorry, 4 of them. You can make a sucrose. You can make amino acids, you can make nucleotides, fatty acids, glycerols, whatever you want to make, you can start with PGAL. Then, what the plant does is take the PGAL and bring it back around to make RUBP that can then be used to fix more carbon. Now, you've probably noticed already this doesn't make a whole lot of sense. 3 carbons can't become 5 carbons. You have to go from 3 to 3. So to get around this, we have to do the cycle in a slightly different way than what we've presented here. We have to do some accounting tricks to try to make the math work so the chemists are happy. Okay? And that's the Calvin cycle. The Calvin cycle starts here. Instead of with 1 RUBP, it starts with 3 RUBPs. 3 RUBPs is 3 times 5 carbons, or 15 carbons. We have 3 molecules of CO2, which have 3 carbons. 15 plus 3 would be 18 carbons organized as 3 6-carbon molecules. 3 of these plus 3 of those gives me 3 of these. But it's unstable, right here. It's dotted line, unstable, intermediate. It falls apart immediately into PGA. 3 6-carbon molecules gives me 6 3-carbon PGAs. 6 3-carbon PGAs. I add 6 ATPs and 6 sets of electrons from NADH, and that gives me 6 molecules of PGAL. 3 5s plus 3 3s is 18. That's 3 6s, that's 18. That's 6 3s, that's 18. That's 6 3s, that's 18. Then I'm just going to drop one of these out, use it to make sugars, starch, amino acids, fatty acids, etc. And that leaves me with 5 3s. 5 3-carbon PGALs can be reorganized with a little bit of energy into 3 5s. So 5 3-carbon PGALs gets converted to 3 5s, while 5 3s is 15 carbons and 3 5s is 15 carbons. So the Calvin cycle is meant to show that you can use pGal to make REVP. All we have to do is balance it so that the carbon numbers make sense. Okay, all right, so far? All right, now, this also suggests that if a pGal is used to make everything in the plant, it's being used to make everything that's got carbon in it, which suggests that any molecule that has carbon within the plant, the carbon that's in that molecule arose through carbon fixation. The plant doesn't make carbon molecules any other way except through carbon fixation. So that means that all 3 of these carbons that are in PGAL arose because of 3 separate carbon fixation events in the past. That is, all of the carbons in all of these molecules ultimately came from CO2 being attached to RUBP. Every single carbon in the plant comes from a previous carbon fixation event.